somebody gets me.
Lily, do you know Mrs. McCarthy?
Okay, so we're thinking now, guys, to stop slurping and then get the scooper and try to scoop some of it.
I want to see the scoop of science.
Suction's off.
Okay, where does it get scooped into?
N times 0057.
One of the biobox chambers.
Great.
I don't know which one.
Okay, so before we move, we're going to slurp some of this red sediment. So we need to change.
Wait, we're slurping or scooping?
We're going to continue to slurp since we're here, but we're going to do the red-orange sediment.
We should
want to change?
Flush, right?
Yeah. Oh, yeah. Let's flush first
Roger.
before starting the red.
Flushing now.
And you switch the
119?
container?
No.
Yeah. Okay.
Okay, this next sample will be 119.
All right, and we'll get the red-orange sediment, please.
All right, we have flushed, and we're going to, which jar would you like?
Let's do number 2, please.
Jar 2.
Thank you very much.
That's 2.
Going for red?
Yes.
Yes.
All right, suction zone.
Are you going for the near red right there?
Maybe.
I'll keep it off until
Whatever
you're
you can get.
Oh.
Is he being shy? He like turned around like, oh.
It's like, no, I don't want to do it.
I don't want to sleep.
You
We really
it so
are.
know when you're ready.
We
Ready.
really do have the Teletubby
Suction's coming
vacuum.
out.
It has so much personality.
Suction's on.
Lily, you have Grandma Betty and Jerry tuning in. Tuning
Really?
in.
Hi, Betty and Jody. Thanks for watching.
Off.
I'll have you know that she just waved at the TV screens in front of her.
No, you can't see me. Oh,
Okay,
Nice.
Nope, didn't want that.
shook out the rock.
On. Back on.
on. Yep. You want to let it settle, see what we got?
Does that
Yeah,
crab
that
only have
sounds
one
good.
arm to the left?
Suction's off.
Yep.
Looks like it.
You got in some sort
It's
of
growing
battle.
it back, but it is
It did.
tiny.
Crabs are aggressive with each
Crabs
other.
are aggressive.
One time I was snorkeling and I got to there was a damselfish and they had its nest. They kind of just looks like a blotch of like purple paint almost up on a rock, but the male guards the eggs and they're very territorial. And I was swimming by and there was an octopus and he went in the little crevice where the nest was and the damselfish was really mad. So he kept biting the octopus's arm and then the octopus would take one of its other arms and kind of like slap it in the face. And I like just hovered over him for like 30 minutes and they just kept going at it. Finally, the octopus left and the damsel won. It was quite entertaining though.
At my aquarium, we had a huge tank. There was a cancer crab project going on. So we had this huge tank of like 20 or so cancer crabs. And it was just a circular tank about 1 scot in diameter. And
It never gets old.
it doesn't get old. And if you would open the lid of the tank, all the crabs were like equidistant spread around the perimeter of the tank, like just ready to go. And when you open the tank, it would be like a distraction. So always one of them would go and attack another one.
Oh,
And it was like a cage match of crabs.
Wow.
Did you teach them how to cuddle? Like a shrimp?
No, I did not train the crabs.
Little did they know.
A shrimp startled me one day, and I punched the tank, though, on accident. I was cleaning it, and the shrimp, like, jumped out, and I went like this, and, like, punched the tank in front of me.
So don't take Lily to haunted houses.
Can we dim the light on the jar to see if we can see
You
how
could
much
turn off some
sediment's
lights,
on.
Yeah,
right?
It's just so bright we can't really
See.
I
tell.
can kind of see a little bit of an edge on the bottom right of the jar.
Oh, barely,
That'd
yeah.
be downlight, maybe? No?
I think it's just either way, it's going to be hard.
Oh wait, maybe. Well, yeah.
Maybe we just get more to be safe.
Yeah.
Yeah,
No,
let's
it's
just
like
get some more to be safe.
Just
It's
blown
hard
out.
to tell.
Yeah, any of that darker red would be great.
Okay.
Oh, to the bottom right.
Ready for suction?
Yep.
Suctions go.
Suction's on.
Oh, jeez.
Oh.
I
Your
mean-
turn.
Jeez.
I'm going to reposition.
Alright,
It's just
suction's not
awkward.
on.
Look at you churned a bunch up for us.
Yes, I did.
Ready?
Ready.
Suction's on.
Oh, nice.
That crab's coming over to be sampled.
He's like, you're taking my habitat away. Could you tell if any more got in there? Yeah, I can't see.
Give it a blast.
Oh
See if
yeah,
it's
it's
just sitting in
turning
the hose.
red now. Yeah, I think we got some now. Is there a minimum amount that you guys need? Okay. I think we got enough now.
Okay.
Yeah. So we need to try and scoop some white, but there may be other patches. So maybe we look for a area where there's the black and white, and we can land and do those last two samples, and then after that, we'll be off the bottom on our way up.
So wait, are we, just to clarify, are we picking up at 6 or are we
I think
picking
you're
up at
hitting the
4?
camera.
Chris, yeah,
Oh,
Chris
shit.
just notified me. After we get these last two samples, we're gonna
Thank you.
head on up.
Okay.
We're ending early.
What the
Yeah.
hell is this?
Oh, the weight on the bottom is kind of nice. All right.
Did
so
you
if
want to move to
guys-
another location, Scott? Maybe somewhere there's a bigger patch might be easier?
Yeah.
If you guys are going to collect these guys, it's going to be, the only open boxes we have are in starboard, so C and D are open. I don't know if that's going to be kind of difficult, moving a scoop around.
A little bit.
I
So
think you
this-
can do it, Scott. I
Thank
have
you,
faith
Scott.
in you.
I will do my best.
This is the Mark II scooper.
Apparently this is the Mark II scoop. What was the Mark 1?
What happened to the one?
Somewhere in the ocean, maybe.
Oh, look at all these crabs. They were like lined up watching the show. On the left. There's a ton of them. Let's see. Chris chimed in and said to go to the east. Okay.
Oh, there's a nice
He
white
was saying
patch. Do you think maybe we want to get it from one of the cracks or like one of the big patches? What do you think?
Yeah, we can do this patch that's right in front of us. We just found this one. Do the white sample right here for the sediment.
Okay.
Okay.
Is Rebecca still on?
She is.
Yep.
Rebecca, we have a question from Holly in Australia.
Chris, do you want us to get a
all
sample
right.
of the sediment to the left that's more black in color too as a separate one?
She asks, do the extremophile bacteria end up facilitating the transformation of these sediments into something that larger, more complex
Okay,
creatures
and we'll get both
can
of them
survive
from right
in?
here.
I'm gonna have to, you're gonna have to repeat that question, sorry.
Do the extremophile bacteria end up facilitating the transformation of these sediments into something that larger, more complex creatures can survive in?
Yeah.
I
That's
thought you were
probably
just
what,
going to end with that.
I thought you were just gonna say
Yes.
short answer. But yeah, that might be something that these crabs are utilizing here. So yeah, the bacteria and the other microorganisms will be able to eat the compounds in the soil and produce different other compounds or metabolites that other creatures could theoretically use, or they might be feeding on the bacteria themselves.
So this looks like black sediment right in front of us at 6 o'clock. Do we have enough bio boxes to do 1 scoop black and 1 scoop white?
We do.
Okay.
Do you know if C and D, because I know for A and B there's a little bit of a separation underneath and water can wash back and forth?
This looks like a good spot. We can do the whiter
Yeah.
one to the left and the black to the right.
We'll just have to do it. Okay.
Yeah, I don't see a way of avoiding it at this point. Unless we want to, because I don't think the scoop itself fits in the bio box.
Oh, so we're going to have to
Nope.
let it
Yeah,
dangle.
that'd be
Might be a little challenging.
a little challenging. Hercules deep sea made. It's like going around with a little scoop and a vacuum. Deep sea cleaning crew.
All right, scooping some white.
Yep, scooping some white. And then what box again was it, Peyton?
That is going to be starboard bio box C for this first one.
Thank you.
And we're on 20
Thanks.
or 19?
20.
This is 20.
Sorry.
You're good.
I think I finally have the numbers right.
Nice.
There was one that I thought I erased, but I just unselected it, so the numbers kept incrementing still.
Ooh.
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 Maybe we could try a bush core.
I don't know if it's dense enough to stay in a push
Yeah, it might
core.
not stick.
It's a nice scoop. Okay.
Excellent.
That would? Chris thinks that it would stick in a core.
Should we try to do a core instead?
That's how we did it.
yeah,
We
try
didn't get very
both.
deep.
Okay.
Ready?
Ready.
So after we dispose of this sediment, we'll do a core as well.
So you said C?
Yes, C is correct.
Charlie.
C for correct, Charlie.
Oh, vertical.
It's
Do you want
gonna
me
be
to
a little tricky.
Yeah.
shut it off?
Take that. Hopefully it don't bounce.
Thrusters. So it's your starboard vertical.
Yeah.
All right, your starboard vertical is off. No, it's not. It's
Oh, it
off
is.
now.
Okay.
Like a glove.
I don't think any of it's going.
Oh, shit.
Well,
I guess
What
it's
was in that
one.
box?
It was a white pagoda rock.
Oh.
Well, it's got white
And
sediment,
sediment.
on it.
Plus sediment. All right, let's try to do. Oh, jeez. Let's try to. Can you make note of that, please?
Sprinkle a little sugar on it.
So let's definitely do a push core for sure now.
So poured all over starboard bio box. Yeah.
It's gonna
Sounded
be
good on
Okay,
paper.
got it.
multiple stab push core.
Yes.
So just
Do you wanna
stab
end that?
away.
Until it's full.
And I'm gonna wait until it's closed.
Oh, okay. Wait, I don't know if they're gonna close it before the push core.
You want to? Yeah,
yeah.
we can.
Oh, okay.
You want to? I can close it temporarily and just bump it out a little bit.
I mean, I think it's not. Yeah.
Looks like it has dry ice
All
coming
right.
out of it.
Oh, we'll bring
It
it in.
really does.
Is that a magnet on the bottom of it too?
Yeah.
Yeah.
Weight? Oh, nice.
Okay. I'm gonna mark closed in 3, 2, 1. Closed. 0, 1, 1, 12. Or 0, 1, 12.
So now the push core.
Going back out.
And then maybe we
Oh,
try a
sorry.
push core for the black sediment as well afterwards.
This is gonna be... I just muted myself when I meant to talk. This will be sample 112. Wait, sorry, hold on. 121. I can numbers today. NA112-121.
Actually, can we use a different push core? This is going to be really picky, but since the other one was used in another spot,
Yeah.
thanks.
I was just gonna say,
I
didn't
was
we
wondering
use
about
that
that.
one?
Or are we using the special sphere?
We are.
Okay.
Anthony, I think your family is willing to send us care packages.
Oh, would you look at that?
Awesome.
Midnight Baker.
Yep. That's what my mom does.
We'll have to get an air delivery because we'll be in transit to a PIA soon.
That's true.
Please.
Pizza delivery by drone.
Yeah, Scott still owes us pizza.
That's
Oh,
true.
This is true. Nice. Oh,
So then he wants it to be.
Oh,
Oh, it's going in
Oh
more.
wow! Hey.
oh wow.
awesome.
It's also
Is that
lifting up the
vertical thruster
ROV.
back on?
Negative.
Okay.
You want it back on?
No.
If you could stab
Ska-
that multiple times
Yeah,
in the
let's-
white.
To the left, yeah.
So normally with the push core, I guess this is also for the viewers, it's very useful for getting the stratification or the lines in the sediment because different microbes will grow in different parts of the sediment depending on what they like to eat and what
Can
they
you zoom
don't
in
like
on
to
that?
eat. But for this case, we're getting the sample and we only care what's in this area. We don't care what's on top, what's in the bottom.
So,
So
the
that-
flapper valve isn't working?
Oh, yeah.
Oh, it's yeah. That's
What if you just sort of flip it upside down?
And then
And then what?
Oh, yeah.
it's like the rubber.
Or
Gonna-
maybe put it sideways as you carry it over to be placed
I mean,
in.
it'll still wash
Yeah.
out.
shove it on that brown rock.
Just get a different one.
Get a different one.
Yeah.
Ready?
Ready?
Okay. You want me to? I can also come out more.
Okay, so white sediments from microbial
I'm
mat.
going to try to straighten it out. It's all
Multiple
crooked.
pushes.
Okay.
Oh, maybe not.
There's
Maybe don't need to.
nothing in that core, so we're gonna try another one.
There's a little bit of...
There's white dust.
Okay.
Yeah.
Oh, crap.
Oh, no.
Ruh-roh.
We'll pick that up later.
Okay.
A little bit of magic smoke. All right. How should we mark this?
Push cores suck.
So you're saying there wasn't anything?
There's nothing
I
in
don't-
that.
Yeah,
I don't-
let's just keep going, and this will be the
Okay,
same
we're
sample.
gonna ignore 121.
Yeah, well, this will just
Yeah.
be like
Yeah,
another.
I don't think there was really anything in that.
Yeah.
I can come out if you want me to.
Yeah,
A
come
little
out.
bit more.
So a great question coming in from the viewer. As we finish and as the ROV surfaces and we get them both on deck, we'll stop answering questions, but you can actually see the team in the wet lab start to collect those specimens, label them, catalog them, do the eDNA, put them in ethanol or in the freezer, photograph, measure, all that good stuff. In terms of identifying them, we try to identify some things now while we're collecting them, but if it's something that's unusual or new that we don't necessarily, or just don't frankly have the capability or expertise to identify them, that's something that happens a bit later on down the road when the samples make it back to their intended destinations. You can continue to check out EV Nautilus at nautiluslive.org, and you can go to latest news in the tab right there at the top, and you can see some of the new things involving the EV Nautilus that have been published. Papers and things like that that might use our information or the samples we've collected can take a little while to make, and they're usually done from a whole host of different scientists and research facilities. I don't think we have a central location for all of the scientific papers that involve the Nautilus samples. But the best thing that you can do is if you go to the expedition and you see all the different expedition years that open up, you can see all the different locations, and you can see a lot more specific information about each one of those, about what was done. Typically there's some pictures and some video highlights and blog posts, so we try to give you as much of an experience as possible.
There you go.
That
That
is
one's
working
working.
beautifully.
Nice.
Very
I'll
nice.
have to make a note of that other one.
Yeah, I took a note about that one.
I'll have to fix it.
Look at all the microbes. So exciting.
Looks good.
I can see them with my eyes.
Absolutely.
I'm glad.
All right.
How many microbes do we have, Rebecca? Can you count them
At
for
least
me?
10.
At least 10.
Oh, sorry. Ready?
Okay, multiple pushes
You want me to switch?
Yeah, please.
of white microbial mat with dark sediment
Yeah.
underneath.
Awesome.
And it all just fell out.
There's still some left.
Still half a tube.
Yeah.
And that's starting to fall out.
Oh.
Are push cores the hardest instruments to sample with?
When they're hanging out on the side of the vehicle, I guess they are. Or when you're trying to do it into loose sediment.
Chris,
Okay,
what'd you
I'm going to
say?
log that there.
Okay.
0121.
What's the term of futility? Something or insanity?
We may want to try to do another one. It looked like it might have been completely empty.
Oh, great.
Maybe.
Go back.
Oh, the one we.
You want to use the same one? Oh, sorry.
No, they're going to make me
Can
grab
we
another one.
pull the box on the starboard
Yeah,
side
could
and
we
then
just like poke it in the sand there, possibly?
What do you mean pull the box?
or
like,
like
just
sample right by the box so it's real quick to
do it on
pop
the
it
side?
in?
I'll try.
I think we're going for another one.
Okay, this is going to be 122. Just
How
going
the
to check
hell are you
that.
supposed to get over
Yes. Last one was 121.
Let's push core number 3, right?
Yes.
You want me to bring it in? Maybe the angle will be better if the box is in more.
there? I have no idea.
I want
I'm
to try
pretty
it.
over it.
Okay.
Maybe I should have said it off SBO.
You can do it.
Now you can. Now.
So for our viewer Kim, we don't have any capability of doing audio from the wet lab. You just get the video there. But you'll get to see all the excitement as they
Hunter, are you going
catalog
to
the
come help in
samples.
the wet lab today?
I would like to, but I have to type up a final report thing for Chris and wrap up some other stuff. But let's see, maybe.
The dirt
Yeah,
is
I
really
gotta catch up
shallow
on
over
some
here. It looks
mosaics.
like we
I'd love
can't
to
really
help
get
you guys,
much.
but unfortunately I got quite the list of stuff still to complete before disembarking.
We're not getting anything in this push core.
Okay.
Do you want to bring it to the front and try again, or move the ROV?
What if we rotate the ROV?
To do what?
So that we're the starboard bio box is facing the soft section where we were earlier.
Got it.
Would that be easier for you,
No.
Scott? No.
What about using the scooper and scooping in this
normal?
side? That was challenging, try to get it in though.
What about a robot with a human brain?
Sorry, Scott.
So the question coming in, we are not going to be going to Ofu Olusegun or Tau anymore. We, after we finish this dive, we're actually going to be transiting back into port for a shift change or personnel change.
That's a crab.
Starboard salvo.
Agreed.
I didn't see that before.
Okay.
We're doing some side sampling.
Is it going in at all? No, huh? No.
A little bit. Oh.
It's all just dumping out.
Oh, yeah. Was there anything in the front left box?
Yes, that was where 117, the white pagoda rock.
Okay.
That was where we, when we had the nice like movie shot with
Okay.
Bob Ballard.
I think there's some sediment in there.
Yeah.
Mission accomplished.
Thank you, Scott.
Thank you, thank you.
Just go rescue a core.
We gotta get one more with a black sediment. Mats.
I need to rescue a core too.
Yeah, and rescue a core.
I'm going to log that now. 0127.
Oh.
Thank you.
Are both thrusters back on?
Negative.
Yeah, we need both now.
Turning vertical starboard on. Vertical starboard is back on.
Thank you.
What if we put it in the scoop and then hold the scoop the entire way up, being very careful not to drop any of it
I
the
doubt
entire 674 meters to the surface?
we would end with a clean scoop.
Oh,
Where'd that
Our
core
knife.
go?
Our life? What?
our knife looks like it's hanging off the side.
Uh-oh.
Awesome.
Oh, wait.
There it is.
There's a core tube in front of you, and
The knife just fell.
I think so.
Where was the knife?
It was just
It
dang.
used to be tied right there.
Oh,
It's
I
not there
see.
anymore.
It's a red thing.
We're losing our marbles.
Yeah. Gone.
It's okay, we can rescue them. We'll rescue them back to their home.
Is this like a metaphor for sanity, or?
Number 2.
Turn starboard vertical off.
Sure.
Was this the one that was broken?
Yes.
I believe so.
Oh,
All right.
It's the dud.
Starboard.
disappointing.
Salvo, you ready?
Yep.
Sample out.
So the second to last push core on the right was empty, is that correct?
Yes,
Okay.
that's number 4.
I'm gonna have to ask you to do one more, Scott, in the black
In the black.
sediment.
Yeah. Right.
And then we'll call it groovy and go up.
Call it groovy.
And retrieve our knife.
Groovalicious.
Oh, and we need to get the knife. Thank you, Brian. Almost forgot.
We'll just like kiss on the head with the manipulator arm.
What's that?
Oh, never mind. Scott gave it a little tap into its hole.
Nice.
Huh. Did they try to do something with this one before? Looked like there was stuff in it.
Yeah.
Not that I'm aware of.
Huh. Oh, maybe. I'm pretty sure.
Maybe it was just some of the material that scattered into it earlier.
Yeah, go for it. Yep.
Is that a worm?
So the black stuff over here?
Yeah, the darkest patch that we can get. So let's see. Maybe.
Do you want to say that over
Was that-
the SPL?
There was a pretty dark patch when we were pivoting to the left before, kind of off in the distance.
Okay. Let's check.
What
There.
about the one right by the lasers?
It's an option. Let's see. Oh, that one right there, maybe?
Yeah,
Yeah, that looks pretty dark, the one on the right.
and it's like the very
just
white.
below the lasers.
Yeah.
That one's pretty dark. Yeah, we could try this one right here.
Your starboard vertical is still off,
Roger that.
but put the tray in, so.
Look like that knife fill kind of just behind us, right?
Yeah.
For future.
Yeah.
Okay.
We have the Doppler track, so.
So we'll do the same, try to fill it up as much as we can.
From hell's heart, I stab at the...
Oh. I was hoping it was going to go more than that.
That would have been a lot more relevant when we were sampling the white sediment, but...
Nice. Well, there's an idea. Haha.
Okay, come up.
Oh, what's that? Nice.
Now.
Slow and steady.
Now what do I do?
I'd say try to get a little more. Keep scooping. I wonder, do we have a bio box that's empty and maybe we can dump? This might be easier to dump into a box than that.
We could put it in
shovel.
Starboard D.
You want to try that?
Yeah, let's.
Okay.
I think that would be easier for you.
Yeah, the scoop just was too big.
Yeah, it was pretty big.
Yeah.
Couldn't really turn it at a good angle either.
All right.
Salvo? Yep.
Which one's open, Peyton?
D.
Okay, the one on the far right.
Should I call this a scoop or a slur?
Top right's bubble.
Where am I?
Top right. Top
more
right.
of a scoop.
All the way right.
Call it a
Oh, that's a good point. Oh, good angle. Maybe see if you can get the tip maybe like halfway, a quarter in the box. Nice.
Was this the black sediment?
Yes.
Correct.
This is 1, 2, 3.
Nice. Come out. Awesome.
There you
Great job.
Scoop core completed.
Scoop core.
Camera.
Woohoo. Oh, there's quite a bit in there. Awesome. Woohoo.
Is that the last thing?
That should be the last thing.
That's it.
Plus the knife.
Now we just got to get the knife and we are good for takeoff.
My perspective's all wacky.
Joe can do it.
Nice.
There you go.
Awesome. Woohoo! How many samples did we get this dive, Payne?
Whoa.
That's going to be a big number.
That's a
1, 2, 3, 4, 5,
Over
6, 7,
14?
8, 9, 10, 11, 12, 13, 14,
Okay.
15, 16.
Put that in.
16 samples.
All right.
Do we need a control Niskin?
I don't think so.
So
Because we're not
You
doing
sound so
eDNA.
sure.
This is true.
Rebecca?
can you zoom in on
Scour
that,
for.
Yeah, I don't know exactly how they
Can
want
you
to
zoom in on
process
that,
this
G?
or what they're looking
Yeah, I
for.
don't
They said
think
they were
they
going to email
Have they
something,
emailed us
Yes.
but probably not. I would imagine they would have asked for a
Is our
control
track,
if they
how
wanted
long is
something
the
else.
yet? Yeah.
track
Okay.
showing
Then
on
let's call
Doppler?
that good.
Just want to like kind of retrace.
Like
We're looking for the
back
9.
Oh,
here.
oops. Sorry, I forgot to log that. Okay, thank
They're
you.
like 3,600 receivables.
Oh, starboard thruster.
Oh, yep, sorry.
That way you're all wonky.
Starboard
Yeah.
thrusters back.
Roger that. Thank you. It's like a blue and red handle. Is that right?
It's red, definitely red, and I think blue tape or something, or red tape and blue.
1.
There we go. Now it's excessively long. So
Yeah,
it should be in this area.
maybe like just 10 minutes worth. I don't know. We'll just have to scour because it's hard to tell on that.
Yeah. Or we just lost a knife.
Could we go higher and get more of like a larger view?
It'll be hard to spot it.
Whoops.
Maybe
You think?
zigzag.
Shouldn't be too hard to find.
Yeah, it was pretty bright red.
Yeah, I don't think we would miss it if we saw it.
Let me look out with bubble. I guess. So the Doppler, we never went that far to the.
Do you want to take the samples with
Yeah,
you?
it's going to be further
So
north.
go.
I mean, I'll take the
north.
whatever.
You don't
extra
think it's
sheets are left
behind us
over,
at all?
but we're not gonna.
No.
Okay.
Our samples were...
Maybe
We were
bearing,
up.
like, yeah, that's a good heading to go back to our middle of our track.
We're sure it actually fell, right?
Yeah, I mean, I didn't
It's
see
not
it.
like just dangling off the side of the vehicle.
I
don't think we technically know.
Okay.
uh,
I mean, it's
We look
definitely
on
not
bubble
there,
cam?
but
Look, there's our footprint last
Yeah.
time
yeah.
from where we landed.
Is that the knife?
Where?
Where?
No, I
That's
was
the
looking on the
scoop.
porch.
Is that the crowbar then?
That's like we have two knives. We have that
Mm,
J one
okay.
and like a regular straight.
Could you see it with Argus cam? If
It's
it's
too.
like dangling?
It's just
Oh.
too. The vis isn't good here.
Mm-hmm.
Yeah, I don't.
Where are you?
Oh wait, right. Look right under Mongo. Like drive forwards
Get low and
and then drive
back.
backwards.
ah,
Is it?
It's
Dangling?
dangling?
Yep, it's
it
dangling
is.
right there.
Oh, I see it.
Do
So we're not going to
Awesome.
find it on the seafloor.
Okay, cool.
Good eye.
Good
and
eye.
I
Well,
know
we found the
and
knife.
it's wedged in there, but if you want to try to move Mongo and get a better grip on it.
I always forget about Mongo.
Yeah, it's still there. Go maybe elbow up.
ooh.
Oh,
What's it holding on
what is
to?
happening?
Spicy.
It's the knot on the end is just jammed on the
On the what?
whatever was supposed to be holding it.
Okay. Cool.
Zoom
We
out,
could just-
zoom out a little bit.
Wedge Mongo's like jaw on it, maybe.
Oh,
I'm gonna
floating.
mark off bottom.
Yeah.
Shit,
Uh,
I'm driving down.
okay.
I was floating.
We're not off bottom.
Oh, we're not off bottom?
No, that wasn't an ascent.
Oh, thought we were
It
up.
was a...
Just kidding. Oh, it's barely hanging on. It got caught or something in the corner.
Nice.
Hmm?
Do you want to put it in a box?
I'm just going to hold on to it.
Okay.
Herc with a knife.
Right. Mission accomplished.
You want to do the recovery?
You had your lesson.
I can share. I went to kindergarten.
Summer likes doing recoveries.
All right, are we marking off bottom now?
Off bottom.
Thank you.
All right.
The true off bottom.
666.4 meters.
So leave hydraulics on. Don't push the boo button. Otherwise, we'll drop the knife.
After all that.
Some crab is going to get that knife and become the most powerful crab.
I feel like that's a YouTube video.
One knife to rule them all.
Can you zoom out on NavG?
That's a good title.
I've definitely seen pictures of crabs with knives.
Yes, yes.
Cool, thank you.
Sorry, I'm writing times down.
They cannot be trusted.
Yes.
All right.
I'm writing times down.
You need to spin and I need to spin, huh? I'm going right.
Aw, Benthic Buddies is gonna dissolve.
Yeah.
Aww.
I'm changing.
Hey, we still have like a solid hour until we hit the surface.
Okay,
Actually,
ready?
Hannah,
Like
I meant to-
maybe half
Out,
hour.
I
What's
meant
up?
to ask you, so what was your sort of path to undergrad, grad, and like where you are now?
Well,
Tell me your life story.
Started my life story. I started in undergrad, majoring in oceanography, thinking potentially to change to studio art to do photography.
Oh,
At one point, I think it was like my sophomore year, but then I decided to stick with the science, and I'm
Yeah.
glad I did. I didn't know what I wanted to do, but I loved the ocean, so I didn't know what particular part of the ocean or area of focus. Ooh, sorry,
hello.
what's up? Are you calling me Lily? Oh, sorry. Okay. And while I was in undergrad, I kept taking classes, and then I did some internships. So I did an internship working with a physical oceanographer in Barrow, Alaska, and I realized I didn't really have an interest in doing that. And then I did an internship at the aquarium in Long Beach Aquarium in the Pacific, which was really cool. But when I also did that internship, I got to volunteer with a group in LA on a restoration project. It was a giant kelp macrocystis restoration project. And when I did that, I enjoyed it a lot more.
yeah.
And I also got involved with doing, I don't know if you've heard of Reef Check. It's an organization.
not.
They do reef surveys, and the survey protocol is very simple, so that anyone, even if you're not a scientist, you can volunteer and help out and collect this data for scientists. So I started doing that and really enjoyed that, and then got really into the restoration stuff and kind of grabbed my interest. And then I had another internship with NOAA. I was a hauling scholar on
You got hauling scholarship?
yeah, so I had the opportunity with that scholarship, with the internship. You do one internship. It was a 2-year program.
Yeah.
Really awesome experience.
I'm planning on applying for that.
You should definitely apply.
Yeah.
It's a really awesome experience. NOAA gives you a lot of exposure. It's great all around.
Yeah.
And so with that, they give you the freedom to intern anywhere in the U.S. or U.S. territories. And the only requirement was that a NOAA employee had to be your mentor. So I was about your age, I think, when I got it, like
Yeah,
19,
you
20,
apply right as you're
something.
going into sophomore
Yeah,
year.
so it's like your last 2 years you would be in the
Yeah.
program, basically. And so I actually tried to come out to American Samoa, but it was just after the tsunami. And so I think it was actually the sanctuary where I'm working now that I had contacted.
Yeah.
But they had to turn me down because of the status of everything post-tsunami. So I kept looking for, I had an interest at that point. I had an interest in, oh, yeah. I had an interest, hold on, let me switch mics.
Hey, Peyton, what number did we get to?
For samples?
Yeah.
1 sec. That would be, I think, 123 was our last one.
123, okay.
And that's the last, I mean, theoretically last sample of this dive, unless we
the
slurp a
cruise.
bunch of fish on the way up. Oh, you're off SPL?
Okay, I'm back. So I had an interest to study tropical marine environments, specifically corals. So that's why I had the interest in the South Pacific. And then after that wasn't an option, I still want to do tropical environments. So then I started looking in the Caribbean. And I ended up linking up with NOAA's Southeast Fisheries Restoration Center out of Florida. And they also
I'm sorry, Peyton. One more time, what number did we get to?
Virgin Islands.
1 sec.
What number?
The last sample was 123.
123?
Yes.
Okay.
1, 2, 3, nice.
Yeah, can you move your mic a little closer to your mouth? There you go.
Is that better?
I think so, yeah.
Yeah, so then for my internship, I ended up going to Puerto Rico. And I did a lot of reef restoration and coral restoration work, specifically with coral nurseries. Fell in love with it, and that's when it hit me, this is what I want to do. So I did my summer internship, and then went back, finished my last year of undergrad. And then while I was in that internship, actually, I had done some networking, and I had went over to the U.S. Virgin Islands for like a weekend. My mentor was doing a vessel grounding workshop, response workshop. And so I had met some of the people in St. Croix who worked with the Nature Conservancy, and they were just starting their coral nursery project. So kind of made those contacts, and then it kind of worked out that just before I graduated, they had a tech position open. And so I applied and I got the job. So then I had like finals on Friday, graduation Saturday, was in the airport like a couple hours after graduation.
Oh my God.
Started a full-time job on Monday.
Oh my goodness.
So I did that.
Wow.
Yeah, it was, it wasn't that long of a contract. It was less than a year. So I was there, I think maybe like 8 months, 9 months. And then after that, I didn't know what I wanted to do. I was looking for other jobs. And while I was looking and trying to find something, everything required a master's or 4 or 5 years of experience, but I just graduated, so I obviously didn't have that. And then
bit of a catch-22.
Yeah, it was hard. So then I ended up deciding to go to grad school. So I had not too like a few months. I think it was like 5 or 6 month gap kind of in between. Applied to graduate school, and then ended up applied to a few different ones, but I ended up getting into University of Puerto Rico and decided to go back to Puerto Rico. I really liked it, so I went over there, and then, which was great because while I was in school, I was able to continue working with NOAA's Restoration Center on some of the stuff I had done previously with my internship.
Got it.
So I was able to continue working with them on a variety of different projects, and that was really, really fun. And then after I graduated, just before I graduated, I moved to the U.S. Virgin Islands. I did some work over there for Restoration Center. I would get contracted out to do some, I would do like vessel grounding assessments if there was any groundings there, because they didn't have anyone based there. I did kind of tap into some of the nursery projects in the Virgin Islands still at that point. I worked for an environmental consulting company, and I also worked for EPA certified drinking water lab. So I was working in the lab there as a, I'm trying to remember what my official title was. I guess lab analyst, was running samples, and then moved to American Samoa, and now I'm a research scientist at the sanctuary.
Awesome.
Yeah, so we're in the process of starting a variety of projects.
Yeah.
We have one going right now, which is an ocean acidification project. We have a NOAA buoy out in our sanctuary management area of Whangatele Bay.
Of that.
Yeah, so we started that, so we're doing bi-monthly sampling there, so that's already going on. And we try to do monthly tows in the sanctuary management areas that we can reach, which is Whangatele, Whangama'a, and Aonu'u areas. And so we'll do tows there to look for any coral bleaching or disease breakouts or crown of thorn breakouts. We're also going to be starting soon a monitoring project, a reef monitoring project in all our sanctuary areas. So we were trying to start this past year, but the funding didn't come through yet. So this upcoming year, in the beginning of the year, we'll hopefully start that. So that's exciting. That way we'll have just an annual reef monitoring project within the sanctuary management area to track changes through time.
Yeah.
And with that project, we're also hoping to use some 3D imaging.
All right, Scott, your life story. We're ready. Give us the origins of Zeus. You're not on the spiel. Everyone needs to know.
All right, sorry.
Oh no, and then another project we have coming up, we're going to be doing a coral nursery project, which I'm super jazzed about. So
that'll be really exciting.
Yeah, super stoked for that. So yeah, so we'll have a coral nursery project starting up very soon. And let's see, what else are we doing?
No, it doesn't work that way.
Yeah, that's pretty. We do have a hydrophone we were going to put out in Whangatele Bay eventually, just to keep an ear out, have an ear underwater, and look or see if we catch any whales. So whales do pass through the Aonu'u management area. They'll pass by Whangatele, but they don't really come into the bay. It's a really small, narrow bay. But dolphins also come in.
Oh.
And Whangatele Bay is one of the management areas that is absolutely no take. However, that being said, we don't really know how, enforcement doesn't really go out there so much. So just because it's a no take doesn't mean that there isn't anyone going there and taking. So the hydrophone too, we hopefully hope to pick up boat traffic in there. Right. We won't know if they're fishing or not, but we'll get somewhat of an idea of how much boat traffic
and if
passes through.
You might be able to find the patterns and then.
Exactly. So, and then Whangatele Bay, so it's to the southwest side of the island. It's pretty out of the way for anybody on a boat. Yeah, so you'd really, it's not like if there's a good fishing spot and it's kind of on the way, like it's more out of the way. So every time we go out there and we're doing any field work, I don't know that we've ever seen another boat. It's pretty much just us.
Yeah.
Yeah, we recently did a study looking, so there's a landfill on the other side of the ridge in Whangatele Bay. And we think there may be potential heavy metals and contaminants leaching through groundwater, but we're not sure. So we just had some scientists from NOAA, I cannot remember which branch or which lab they were out of, but they came down and we did this study for a week filtering water. They had these little instruments that they called clams, and they basically act like a clam.
Oh,
you put the filter and then they can, and it's a new design, and the company that made them, they're still fiddling with them and tinkering with them, but the idea is that they act like a clam, like a filter feeder, so they can, and then the instrument also keeps track of the flow rate. So you know the volume of water that went through, and then you can compare that with how much of whatever you collected. And so we did that for about 5 days.